Introducing Agentic AI orchestration of MD skills, topped with narrated trajectory videos through IQ. The agentic AI skills will assist you at all stages of MD, starting from understanding the problem statement at hand, identifying correct simulation protocols, preparing, running, and analyzing the simulations. IQ visuals will ensure you do not have to spend hours on visualizing softwares to observe the events of interest throughout the trajectory. Please visit Aganitha's IQ gallery to appreciate the narrated visuals generated once the MD skills are done with their jobs. Different agentic AI MD skill packs are available depending upon the scientific context. For example, agentic skills for drug and material discovery, self assembly and permeation behavior in surfactants, peptides, and polymers, lipid and interfacial systems, and drug delivery through LNPs and AAVs. These skill packs can be customized to make your MD agent responsive for the scientific question of interest. When prompted with the right question, the AI harness will call the relevant skill packs, for example, protein ligand binding landscape skills in this particular case. Agents assist you in all aspects of your scientific journey as a mentor, collaborator, and critic. In this particular case of protein ligand interaction, it can help you in asking right questions and answering. For example, what is the correct pharmacophore feature crucial for quantitative and qualitative estimate of protein ligand interactions? What interactions are transient and what are persistent throughout the trajectories? And what is the right conformation of your target of interest when a drug molecule is binding? Is it even relevant for the biological activity? Let's continue with the demo. We started by providing the path to MD trajectories for ligand bound protein. The objective was to understand and quantify the interactions between protein and the ligand. Relevant skill is invoked, files were located, and the agent started by querying the PDB file. It highlighted that the sequence in the PDB file, when compared to the actual UniProt sequence, is showing an offset of 21. Then it moved to basic trajectory analysis to understand fluctuations in protein and ligand through RMSD calculations. The agent will identify frames of higher fluctuations to extract PDB frames for further analysis. Flexible residues and regions of interest were identified through RMSF analysis. Again, the information will play a crucial role in identifying residues and regions of interest for further analysis. The agent is now utilizing the saved PDB frames from the RMSD data to understand intermolecular interactions between protein and the ligand. The protein ligand interaction profiling done over different frames is used to evaluate the interactions that are maintained throughout or were transient and also to identify new interactions that formed during the course of simulation. Using information from RMSF data and protein ligand interactions, the agent was able to identify flexible residues in contact with the ligand. For example, residue 51 in this case. Torsional angle sampling through Janin plots is then carried out. Next, the agent moved to quantify protein ligand interaction strength through MMGBSA calculations. Contribution of individual energy terms to total binding affinity can be traced. Now it is time to have a visual commentary of the entire conversation through IQ. The agent will give you a plan which the user can ask it to modify if not satisfied. Once approved, just sit back to have a audiovisual insights from the analysis. A few applications of IQ to MD simulations are highlighted here. Now let's take a short glimpse on audiovisual narration generated by IQ.
A salt bridge that simply does not let go. Valine 171 in cyan. 17 residues hold this ligand for more than half the simulation, and 4 of them never let go at all. It is the invariant asparagine found in essentially every homeodomain ever solved, and it hydrogen bonds directly to adenine. Change it, and DNA recognition collapses. Factor XA is an enzyme in the blood that helps form clots. It converts prothrombin into thrombin, the step that turns liquid blood solids. So this run scales down the interactions between protein and ligand, an enhanced sampling method that brings the departure into a 10 nanosecond window we can actually watch. Before it goes anywhere, look at what actually holds it. Glycine 206 in magenta makes a hydrogen bond to the inhibitor in about 3/4 of the bound frames. It is the most persistent polar contact in the site, and it comes from the backbone rather than a side chain.
We provide MD skill packs customized to scientific context that works for the harness you prefer, be it Claude, anti-gravity, or any other. Check out our Chem Archive article to know more about IQ. Please visit our website to know more about Igniva and what agentic AI skills we offer. And stay tuned for upcoming demos.